Hi everybody, DrBiology here, and this is part 6 of my biological molecules section of work, and I'm going to look at nucleic acids, DNA and RNA, and hopefully going to look at the similarities and differences. And then in part 7, I'm going to talk about a very famous experiment by Michelson and Stahl. Let's get started. In all living cells, DNA holds genetic information and RNA transfers that genetic information from DNA to the ribosomes. And you'll be talking about that when you do protein synthesis. So both DNA and RNA, they are known as polymers of nucleotides. So each nucleotide is formed from a pentose sugar, a nitrogen-containing organic base or nitrogenous base, and a phosphate group, as you can see by the diagram. DNA, the pentose sugar is called deoxyribose. So in each DNA nucleotide, there's the same sugar and a phosphate group. However, what is different is the base on each nucleotide. So there are 4 possible bases: adenine, thymine, cytosine, and guanine. So A, T, C, and G. DNA is double-stranded and tends to be very long. RNA, on the other hand, contains a pentose sugar called ribose. Like DNA, an RNA nucleotide has a phosphate group and one of 4 different bases. In RNA, though, uracil replaces thymine as a base. RNA is single-stranded and it tends to be very short. So let's have a look at the differences between the two. So you can see DNA double-stranded, whereas RNA has single-stranded. DNA has deoxyribose sugar, whereas RNA has ribose sugar. DNA has the base thymine, whereas RNA has replaces thymine with uracil. And DNA is very large, or it's very long, and RNA is very small or short. Now let's talk about the polynucleotide structure of DNA. So as we know, nucleotides join together by bond formation between sugar of one and phosphate of the next to form a polynucleotide. So the bond formed by condensation is known as a phosphodiester bond. So a DNA molecule, as you'll know, is a double helix. It has 2 polynucleotide chains held together by hydrogen bonds between specific complementary base pairs. An RNA molecule, on the other hand, is a single-stranded polynucleotide chain. DNA molecules exist as a double helix. They have 2 anti-parallel polynucleotide strands held together by hydrogen bonds between complementary base pairs. Now, anti-parallel means one strand goes one way, the other goes the other. The 4 DNA bases, A, C, T, and G, they form base pairs. So A pairs with T and C pairs with G. Okay, that's a whistle-stop tour of nucleic acids, DNA and RNA. So the next video, I'm going to look at the experiments of Michelson and Stahl looking at DNA semi-conservative replication. I'll see you soon.